today we will discuss about the cell and cell cycle and cell division. Yesterday I have provided notes on cell cycle through WhatsApp, and today we will discuss some aspects of cell cycle and followed by discussion on cell division, and here today we will take the mitosis. You all know that first we have an understanding about the cell. What is a cell? What is a cell? How to define what is a cell? We all know that a cell is the structural and functional unit of life. It means that the organisms are made up of cell. The complete organism is made up of cell. That organism may be of human beings, animals, plants, bacteria. Bacteria is an example of a unicellular organism, whereas all other organisms you can take animal, plants are multicellular. So the life or the complete body of an organism is made up of either one or a number of millions of cells. So we call that the cell is the basic repeating unit or basic structural unit of an organism. And why we call it that it is a functional unit? Because every life process takes place in cell. Whatever the process of life we take, such as metabolism, cell division, glycolysis, and everything, absorption of nutrients, that takes place in cell. So the cell is the functional unit of life or organisms. And in 1839, there were three scientists who proposed a theory before us, and that is known as cell theory. Cell theory. There were three scientists, namely Sliden, Swan, and three scientists were there who proposed a cell theory in 1839. And what is in that cell theory? There are three important aspects of cell theory. One is, as we have already discussed, the first point is all living organisms are composed of one or more cells. Then living organisms are made up of cells. The second component of cell theory is the cell is the basic, we have already discussed, the cell is the basic structural and functional unit of the organisms. And the third, today our focus is on this third aspect, third point of this cell theory, that is a cells arise from a pre-existing cells. What does it mean by this? It means if we have this cell, it means definitely this cell has a mother cell. It means only the cell we have, if there is a cell, then only new cells can be prepared or new cells can be obtained or new cells can be formed. That cells arise from a pre-existing cell. It means it is not possible to have a cell, to produce a cell without having a cell. It is not possible. So this third aspect is most important, that cells arise from a pre-existing cell. And the cell cycle and cell division here is completely focused around this third aspect of cell theory. And now we will discuss about cell cycle. We know that a cell is produced by a pre-existing cell. So how this mechanism takes place? This is the process of formation of one cell from a pre-existing cell. This is the process of formation of one cell by a pre-existing cell. And this complete process, when a cell prepares itself to produce new cell, it undergoes a number of changes. Various activities takes place in a cell. This cell prepares itself for the production of new cell, and that comes under cell cycle. So here we discuss it through a diagram. Suppose this is a cycle, and here we can divide this cycle into 4 parts, and starting from here, this is G1, S, G2 and M phase of cell cycle. It is represented, cell cycle is represented by this diagram. So what is the significance of all these steps? Two steps are most important. S phase. S means synthesis. Here, what is synthesized? Because if this particular cell has to divide, then this cell undergoes a number of changes. But before division, the cell synthesizes its genetic material. It means whatever the DNA or genetic material present in this cell, which is undergoing cell division, every component or sum total of its all genome or every material, every genetic material that is included in this cell has to be doubled. So here we know that process is DNA replication. DNA replication takes place in S phase, that is synthesis phase. Here, if we have one chromosome, this chromosome will prepare its identical copy, and that's why these two are the same chromosomes and both are the same copy of one another, and both the chromatids are known as sister chromatids, and these sister chromatids remain attached at centromere along the entire length of the sister chromatids. So this DNA replication needs a number of various types of preparation, such as it needs some proteins. We all know that DNA replication, DNA helicase, single strand binding proteins, topoisomerase, a number of proteins takes place before this particular chromosome duplicates. So all that preparation takes place in gap 1 phase. Whenever the required materials are prepared, the cell undergoes for S phase. And here, after completing all the process, all replication of its total genetic material, the cell undergo for M phase. But before that, this cell make it sure that everything has replicated completely. If any if there if any missing is there in DNA replication, or a particular part of DNA is not replicated completely, so it will cause a genetic defects in daughter cell. So a cell maintain or assures that every part has been completely prepared, completed in a successful manner. So that cell checks itself whether the DNA has replicated properly or not, and after the cell assures it that the everything has been finished completely, the cell undergoes for M phase, and this is the M phase or the cell division, where the cell divides. And two types of cell division we know, that is mitosis and meiosis. So the cell cycle has four phases: G1, S, G2 and M. At M phase, which is the smallest phase, shortest phase of the cell cycle, and S phase is the longest phase of cell cycle. A cell cycle takes nearly 18 to 24 hours for its completion. And now we will focus on the M phase. This cell cycle has can be divided into two phases further. This G1S G2, G1S G2 is known as interphase, is known as interphase. So a cell cycle has two parts: an interphase and a cell division phase. A cell cycle has two parts: interphase and M phase, or mitosis or meiosis. So and interphase has G1 as G2 phase. So a cell cycle is completed by G1 as G2 and M phase. So interphase and M phase. Now we will focus on the M phase, that is the cell division. Cell division. And here we will take the example of mitosis. As you know, if this cell has to divide, it has a cell membrane. In plant cell, it may have cell wall, and this cell is now ready for division. You all know this cell has definitely doubled its DNA. Genetic material has become double, because if this cell has to divide into two daughter cells, the total number of genetic material, that is this is a diploid cell, so whatever the genetic material this cell contains has to be passed equally in the daughter cell. So this type of cell division is known as mitosis, when a mother cell gives rise to daughter cell, and the total amount of genetic material is equal, same as the genetic material present in mother cell. If there are 4 chromosomes in mother cell, so the daughter cells will have all 4 chromosomes. 4 chromosomes. So we will discuss about the various steps of mitosis, and it's very important. So before a cell go for mitosis, this cell is known as interphase cell. Interphase, that is the phase before cell division. Here the cell has a cell membrane. It may have nuclear membrane. These portions are nuclear chamber, nuclear pore complex, and here we will have a thread-like structure that is known as chromatin. This is the interphase cell. Now this cell has to go for M phase, and we know that if this is mitosis, it will have 4 phases: prophase, prometaphase, metaphase, anaphase, and telophase. One by one we will discuss about the various steps of mitosis. So the process of this mitosis or equational division completes in these 4 or 5 steps: prophase, prometaphase, metaphase, anaphase, and telophase. So as you know, this is an interphase cell. Now this cell go for prophase. So what happens in prophase? The chromosomes present in this nucleus starts condensation. These chromosomes becomes thick. Here we cannot separate or count chromosomes because this is a thread-like structure known as chromatin, and this chromatin is made up of genetic material, DNA and RNA. So here in prophase, these chromatin fibers starts condensation. So if this interphase cell has 3 chromosomes, suppose we take an example of 3 chromosomes, so this cell has to a number of 3 chromosomes. Here the condensation of chromosomes takes place. So, and chromosomes are visible only when they are condensed, they are thick. So in prophase, and we already know that this cell has passed the S phase. So what happened in S phase? That whatever the genetic material present in cell, it has become doubled. It means if there were initially 3 chromosomes in this cell, all chromosomes has prepared its identical copy in S phase. So we represent the identical copy of sister chromatids in this fashion. So these are the 3 chromosomes, and every chromosome has prepared its identical copy, and both the identical copy of the chromosomes remain attached at the centromere and along its entire length by a protein. A particular protein takes place here, cohesin complex. These 2 sister chromatids remain attached along its whole length and with the help of a cohesin complex protein. And now this is the prophase. The another step is the prometaphase. What happens in prometaphase? Chromosomes start condensation. Now the nuclear membrane disappears. Nuclear membrane nuclear membrane disappears in prometaphase. Now what happens at metaphase? As you know, now we are discussing about metaphase. As the name indicates, meta, it means all condensed chromosomes comes at the midline of the cell, that is at the equatorial plate of the cell. Equatorial plate of the cell. So all chromosomes, and here we took the examples of 3 chromosomes only. So all these chromosomes with their identical copy or with their sister chromatids align at the sister align at the equatorial plate. Now the spindle formation has already started in the before the prophase condition. Spindle formation starts by the centromere or centrosomes, and sorry, this has to be included inside the cell because this is the cell membrane. These centrosome forms spindle formation, spindle fibers. Spindle fibers are proteins, threads of proteins, and these centrosomes moves towards the opposite pole, and both the centromere forms spindle fiber. If this upper centromere form this spindle fiber, if this spindle fiber attaches with this particular sister chromatid, the lower centrosome will form the spindle fiber that will attach with the another sister chromatids. Some spindle fibers are without the attachment to the any of the chromosomes, and so one copy of a replicated chromosome attaches to the one pole, and another chromatid, another sister chromatid with the another spindle fiber at another pole. So what happens after the assembly of this spindle fiber formation? This spindle fiber contracts. These spindle fibers reduces in its length, and this sister chromatids get separated. But the question is, if these sister chromatids are attached with each other with the help of cohesin complex, so how these sister chromatids are replaced from each other? It is all about by the APC protein, that is anaphase promoting complex, anaphase promoting complex. What this protein does, this protein removes the another protein, securing, securin, securin protein, because this securin protein is a part of cohesin complex. So cohesin complex is the protein necessary for the condensation of chromosome, and this cohesin complex has one part, that is securin. So the anaphase promoting complex is another set of protein. This APC complex inactivates this securin protein, and when this securin protein remains no more functional, then an enzyme and another enzyme or another protein, that is separase protein, separates both the sister chromatids, and both the sister chromatids are pulled towards the opposite pole by the continuously reduction in the size of this spindle fiber. So at anaphase at anaphase at anaphase what we observe that one copy of each chromosome has moved to one pole and another copy at another pole. Now here the anaphase process completes, and at telophase process the nuclear membrane starts synthesizing, and right from the beginning of anaphase up to telophase the cytokinesis starts. That is, both the daughter cells start separating from each other. So this happens here in mitosis.